Thanks for the invitation. It is a real pleasure to really have the opportunity to discuss with you some of the work that we are doing, basically supported by one of the AVI centers, the so-called Center for Antivirals, Medicines and Pandemic Preparedness. So basically, the reason why we are interested in targeting mammarenaviruses is because several of them are important human pathogens. So we have Lassa virus, it's basically the so-called underrated human pathogen in Western Africa. There are a number of South American hemorrhagic fever mammarenaviruses, including Junin, Machupo, Chapari. So they are in different countries in South America. And now it's worth to mention that the globally distributed prototypic mammarenavirus, lymphocytic choromeningitis virus, besides being a widely used and extremely highly tractable experimental system to study virus-host interactions and associated diseases, there is increasing evidence that it's also a neglected human pathogen with relevance in congenital infections. And also, LCMV is a real threat to immune-compromised individuals. And this was dramatically illustrated by the fatal cases related to transplantation. So individuals that received organs from a healthy donor that was infected with LCMV, due to the immune suppression, the virus was out of control and basically was a fatal infection. And we have had several clusters of this. And also, in the last 10 years or so, there are a number of mammarenaviruses that they have been discovered, in many cases using metagenomics, and subsequently studies have found that for some of them there is an association with human disease. An example is a Wensu virus that was discovered in China and is linked to respiratory and febrile diseases in humans. And there is a case that happens a while back it was 2008, and it's a virus called Lujo virus that was identified in Zambia. The index case was in Zambia, and that person was moved to South Africa for treatment, and during the treatment, the patient infected another 4 individuals, healthcare workers, and out of the 5 cases, 4 of them were fatal. Now, the interesting thing about Lujo is that we don't know the reservoir, and we never have seen it again, but it illustrates the possibility that there are a number of mammarenaviruses that in any given time can really, via zoonotic events, infect humans and cause disease. As I indicated, Lassa is a so-called underrated pathogen in Western Africa, and I just gonna give you a kind of brief summary about why Lassa really has a significant impact in human health. It's endemic to Western Africa with a very large population at risk. The estimates are that there are over 500,000 infections a year, because it still is difficult to know the ratio between infections and disease. But because there are so many people infected, the number of Lassa fever cases as the disease that Lassa virus causes is very high. And this is estimated at about between 5,000 to 10,000 deaths yearly. In addition, Lassa fever survivors, very often, they have long-term sequelae, including the so-called sensorineural hearing loss. So basically, as the recovery from the clinical symptoms, they lose hearing, and that could last for years to the whole life. Currently, there are no FDA licensed vaccines, and the only therapeutic intervention is treatment with the nucleoside ribavirin, for which the efficacy is highly questionable. And Lassa has really a potential for zoonotic spillover and spread in humans. So this is just basically a brief description of the genome organization of the mammarenaviruses and the cartoon with the particle structure. So these viruses are envelope viruses with a bisegmented negative strand RNA genome. So the genome has 2 different segments, the so-called S or small and the large or L. And each one uses the so-called ambisense coding strategy to direct the synthesis of 2 different polypeptides. The S segment encodes for the virus nucleoprotein and the glycoprotein precursor GPC. GPC is co- and post-translationally processed to form the GP complex that decorates the surface of the particle and is responsible for receptor recognition and cell entry. Whereas the nucleoprotein is the most abundant protein in the particles and in infected cells and really encapsulates the viral genome. The L segment encodes for a very large protein, around 200 kilodaltons. It's a typical RNA-dependent RNA polymerase, or L protein, and a small protein called Z that is a matrix protein. So what happens is that these viruses, negative RNA viruses, negative strand RNA viruses, they form a ribonucleoprotein core that then is linked to the surface glycoprotein via the matrix C protein. So this is very similar for all these envelope negative strand RNA viruses. And now, over the years, we have been developing a variety of tools and platforms to really understand how these viruses work, and also to identify and develop potential inhibitors of virus multiplication. And one of the things that we have been very invested is in targeting components of the Mammarenavirus replication and transcription machinery. As I indicated, so the way that these viruses replicate and transcribe their genome is they form first a so-called nucleocapsid template, is the viral genome RNA tightly interacting with the nucleoprotein. To that, the polymerase associates to form then the functional viral ribonucleoprotein that is responsible for directing the biosynthetic processes of RNA replication and gene transcription. Therefore, if you target the formation of the nucleocapsid, you will inhibit early events of virus replication gene expression. Also, interestingly, as other Bunyaviricides, the Mammarenaviruses, they use cap snatching for transcription initiation. This is like an influenza virus. So what happens is that the virus polymerase has two different activities: a cap binding activity that allows the polymerase to bind to the cap structure present at the 5' of the cellular messenger RNAs, and then the L polymerase has a endonuclease activity that cleave those messenger RNAs, generated the so-called cap primer, and this is what the polymerase uses to initiate transcription. So therefore, if you target the cap binding or the endonuclease activity of the virus polymerase, you will inhibit also transcription of the viral genes and production of proteins, and therefore virus multiplication. So we wanted to really have a system where we could identify inhibitors of the activity of the Lassa virus ribonucleoprotein complex. The problem with Lassa, the use of live Lassa requires BSL-4, which complicates logistically all the process. So what we did is we engineered a cell line that basically recreates or intracellularly forms a active Lassa virus ribonucleoprotein in the absence of infection. So basically, this system, the cells, they express the polymerase and the nucleoprotein, as well as the so-called mini genomes. So a mini genome, what it means is you have all the cis-actin regulatory sequences, but instead of expressing viral proteins, you are expressing reporters genes of interest. And this case is CS green and Gaussian luciferase, and also seosign for selection. So basically, we have done extensive QC of these cell lines. We have done the same thing for LCMV, and the results of the QC confirm that they really work as is expected. Basically, if you use ribavirin or favipiravir, it's T705, or a inhibitor of pyrimidine biosynthesis, that they are all of them are expected to inhibit virus replication gene expression, is what you see. Basically, you can, in a dose-dependent manner, inhibit the activity of the Lassa or LCMV, ribonucleoprotein activity. Now, if you use a compound called PF429242 that is an inhibitor of the processing of the GPC, it doesn't have any effect on the activity of the ribonucleoprotein, whereas it can inhibit the infectious virus. Because what happens, the activity of the ribonucleoprotein doesn't require the glycoprotein, and therefore a inhibitor of the GPC processing doesn't have any activity. So over the years, we have used this system to screen a variety of libraries, and we have collaborated with Caliber to screen several libraries that they have in-house. One is the so-called Refrain library. It basically is a library that put together all FDA-approved compounds, as well as compounds approved by the Canadian health system and the European community and Japan. So basically, this allows you to very quickly identify potential hits for which there is significant amount of information in terms of safety and PK and PD, and you can move much faster. They also have a proprietary antivirals libraries that we have screened and a nucleoside analogs libraries. Using these different libraries, we have identified a number of direct acting antivirals as well as host targeting antivirals that they have potent activity against Mammarenaviruses. And in the context of the AVI programs, we have been focusing in the direct acting antivirals. That is the key themes for these programs. And I'm going to really give you some examples. So one of the compounds that we identified is baloxavir. Baloxavir is an inhibitor of the endonuclease. Actually, baloxavir marboxil is a FDA-approved drug that was developed against flu. And what it does is inhibit the endonuclease activity of the PA protein of influenza virus, and is given to patients. The problem with baloxavir, marboxil, and flu is that the virus very quickly can generate resistant viruses to baloxavir. Now, we found the baloxavir has activity against LCMV and Lassa, but the EC50 is relatively high compared to the one for flu. It makes sense because baloxavir was developed against flu, not against Mammarenaviruses. So in the case of flu, the EC50 is single-digit nanomolar. In the case of LCMV, Lassa is low micromolar. So, but we use baloxavir, in collaboration with Calibre to do an extensive medicinal chemistry SAR campaign that we are not going to go into the details, but basically the overall approach was to introduce an additional carboxyl group into the metal chelated moiety of baloxavir, and also to incorporate into the morpholine moiety of baloxavir a number of cyclic structures of different sizes. And that resulted in a number of baloxavir analogs with high potency against Mammarenaviruses. And I'm going to just talk briefly about our lead compound. Is something called NCPV916. It has very good potency against LCMV, Hunin, and Lassa. And as you can see, the EC50 really, compared to baloxavir, is much lower. Basically, you start to get now a compound that really has potency in cell-based assays. All these are cell-based assays, dose response with infection. And the toxicity is very good because basically it's definitely higher than 10 micromolar. And the data that we have now is in some cases over 100 micromolar. Now, one thing that is important to mention is that this compound, NCPV916, has the ability to inhibit very strongly the virus in a so-called multi-step growth kinetics. Right now, what you do is you infect cells a low MOI, and then you allow the virus infection to go for 72 hours. And in the case of vehicle-treated cells, as you can see, you reach peak titers in the range of 10 to the 6. You're talking about 10 to the 6 infectious units per ml. It's very, very high. And as you can see, when you go over 60 nanomolar concentration of NCPV916, you can inhibit 6 logs of viral infection. So this is not just a AC50. This is knocking down 6 logs of viral infection, what really is really very potent. Now, before we dedicated additional efforts to develop further NCPV916, we wanted to know whether we were on track in terms of in vivo efficacy. So what we did is we used a mouse model of LCMV lethal infection. It's the so-called FBB mouse. FBB is a strain of mouse that if you infect those mice with LCMV, relatively high dose, 210 to the 6 intravenously, IV, these mice around day 6 start to lose weight, and then around day 8 to 12, all of them, they succumb to infection. So it's really 100% lethal. As you can see, if you treat the mice with 30 mg per kg, this is done every day, QD, with MCPV916, you achieve 100% protection. So basically the vehicle treated, all of them, they succumb, in this case before day 10. Those treated with the MCPV916 baloxavir analog, all of them, they survive. And that correlates with the fact that whereas in the vehicle treated, you see high viremia at day 8 post-infection, in the treated ones, you cannot detect viremia. So basically what it's telling you that the compound is able to prevent virus multiplication. Importantly, at the experimental endpoint, only with the mice that they survived, that they were treated, we analyzed different tissues just to be sure that the treatment with NCPV916 not only protected from death and prevented viremia, but also didn't allow viral persistence in different organs, either by titration of infectious virus or detection of viral RNA via RT-qPCR. We couldn't see any signs of persistent infection in these tissues. So indicating that treatment with NCPV916 indeed clear the viral infection. Now, we wanted to confirm that NCPV916 is targeting the endonuclease activity of LCMV, Lassa, and other Mammarenaviruses. The problem here is that there is not a reliable biochemical assay to measure the endonuclease activity of Lassa or LCMV. This is intriguing because for other Bunyaviruses, such as Crimean Congo, La Crosse, there are many of them for which you can purify either the L protein or just the endonuclease domain. And biochemically, in a test tube, you can show the endonuclease activity. However, we and others has failed to do the same thing for Lassa and LCMV. The reasons for that we really don't know. We don't understand why, in the case of Lassa LCMV, we cannot do that biochemically. Now, to do that in the context of infected cells is not feasible because in the case of Mammarenaviruses, replication and transcription are linked. Basically, transcription is needed to produce the MP polymerase. If you reduce level of transcription, you will have less MP and less L, and then you will affect also replication. And if you affect replication, you will have less template to produce the transcripts. As you can see, being linked is very difficult to really assess whether MCPV916 was targeting mainly transcription or replication. To overcome that, we use a cell-based mini-genome assay. In this assay, you can disentangle production of the transacting factor, nucleoprotein, and polymerase, because you provide those via plasmids, from the replication and transcription of the mini-genome. And that is illustrated in the cartoon in the slide. Basically, right now, you're providing the MP and L via plasmid, so you basically can then follow the activity of the ribonucleoprotein that is formed intracellularly in those cells, following the reporter genes that are expressed by the mini-genome. And basically, because the sizes of the genome and anti-mini-genome and the messenger RNAs produced by the mini-genome are different, you can separate them by Western blot, by Northern blot, and then you can quantify the specific species, either replicative species or messenger RNAs. And indeed, what we found is that in a dose response, NCPV916 inhibited more potently transcription than replication. So then, in addition, to further confirm the target ID, we selected for a resistant virus. So we did serial passages in the presence of increasing concentrations of the compound, and we ended up selecting a virus, this one, that is resistant, or has a much higher resistance to the compound compared to the parental virus. We sequenced the virus and identified one mutation in the polymerase, and then that mutation was tested again in the context of the mini-genome. And what we observe again, that basically the MCPV916 basically is much more effective against the parental virus than against the resistant virus. So indicating that that mutation really drives resistance. So we also did basically some binding assays using basically differential scanning calorimetry, and we confirmed that the wild-type Lassa binds very well to the compound. By basically what this assay does is when you have the polymerase with the compound stabilizes the polymerase and increases the TM, whereas the mutant still retain very good TM, suggesting that the mutation doesn't prevent binding to the polymerase, it's preventing the activity of the endonuclease. Now, one of the things that we do not understand very well is that the mutation at 107, the serine to asparagine, is outside of the predicted active site that we actually now have identified via X-ray crystallography. Now, whether this is a allosteric interaction or other mechanisms, we don't know yet. But now what we are doing is selecting a number of resistant viruses to have a complete landscape of the different residues that the mutations in those residues can confer resistance to the compound. And that will provide us with a much better idea of how that happens. Now, one of the screens also identify a number of compounds that they have, that they were nucleoside analogs with a very strong activity against Mammarenaviruses. Two of them are sangibamycin and 4-fluorodiene. 4-fluorodiene is a very well-known broad-spectrum antiviral that is being used now against different viruses and is moving into the clinic. So what we did is we used 4-fluorodiene as a starting point to carry on a comprehensive SAR campaign, which resulted in this nucleoside analog NCOT466 that is active against LCMV, Lassa, and Hunin, and again, in a multi-step growth kinetics, is very potent. It can inhibit more than 6 logs of virus infectivity at very low concentration, 100 nanomolar. So we have a nucleoside analog with a very strong activity in cell-based assays. And also, we confirmed that this compound, NCOT466, is targeting the activity of the polymerase. This was done in collaboration with Matthias Goret at the University of Alberta. His laboratory has developed a number of polymerase assays for different viruses, including Lassa and LCMV. And the interesting thing is that NCOT466 is not behaving as a typical change terminator. So what happens is that when NCOT466 is embedded in the template, is what prevents synthesis of RNA by the polymerase. So it's interesting because this is similar to the mechanisms that Matthias identified for remdesivir. So it's not a classic change terminator. It's basically a compound that one is embedded in the template, prevents the polymerase to use that template efficiently. So, and it's very potent too in the biochemical assay. And importantly, the compound also has in vivo efficacy. So in this case, we use a prodrug, NCOT A14 is the prodrug of NCOT466. It's just to allow for the right plasma concentrations to be sustained over the time needed to prevent virus multiplication. And in this case, we use 2 different models of infection. We again use the FBB mouse model of LCMV lethal infection. And as you can see, there is a dose-dependent protection. When you are 30 milligrams per kg per day, you achieve 100% protection of these mice. That again correlates with the lack of viremia. You cannot detect viremia in the mice that they have been treated with the NCOT A14. And also, as we discussed with NCPB 916, and the experimental endpoint, basically in this case, we decided 12 days post-infection because all the vehicle treated already succumbed at that point. We could not detect any virus, viremia, and also analyzing a variety of tissues, we could not detect any infectivity in tissues or viral RNA. We also tested the NCOV A14 in a mouse model of Hunin lethal infection. This is a different model. In this case, these are B6 mice that they express the human transferrin receptor. That is the main receptor being used by Hunin. And as you can see, again, in a dose-dependent manner, NCOV A14 has significant protection. It's not 100%, but it's similarly potent as favipiravir, that is the benchmark being used in studies to look for antivirals against Hunin. So basically, these mice it's about 80% protection at 30 milligrams, at 60 milligrams per kg. So, and that correlates also with the mice not losing weight, whereas the vehicle control treated mice and infected with hunin, they lose weight and they don't recover. Now, NCOD 466 is also potent in different cell types. This is important because nucleoside analogs, sometimes they have different activities in different cell types. And what we did is to test MCOD 466 in hepatocytes, endothelial cells, and macrophages. The reason for selecting these cell types is because these are main targets of Lassa virus during infection in humans. And as you can see in all cases, MCOD 466 was very potent, reducing peaks titer by several logs. In this case, about 4 logs. Here, 5 logs, 6 logs, 6 logs, 5 logs, and 4 logs. So basically, the compound really, NCOD 466, is active in cell types that they are critical during Lassa virus infection in humans. And one of the concerns with nucleoside analogs is the mitochondrial toxicity, because some of them are being used as a substrate by the mitochondrial polymerases, and that is a concern. What we have done to explore this is we use this Seahorse XF assay that basically what it does is measure the overall physiology of the mitochondrial. And when really we tested NCOD 466, oh, sorry, this is not right. When we tested MCOD 466, we couldn't see any significant effect of the compound up to 5 micromolar, that is far above the EC50, in the basal respiration, ATP production, or maximal respiration of the mitochondria. So basically, it suggests that NCOD 466, at least during the exposure that we did in this experiment, is 72 hours, it doesn't have any significant toxicity in the activity of the mitochondria, which really is very useful for the further development of this nucleoside analog. And finally, because MCPB 916 is targeting the endonuclease activity, whereas NCOD 466 is targeting the polymerase activity of the same protein, the L protein, but there are two different activities. We explore whether these two compounds, they have synergistic activity in terms of antiviral activity. And indeed they do. They have very potent synergistic antiviral activity. We basically did the typical A by A matrix with different concentration of each compound. And then we analyzed the matrix using the Synergy Finder software and using different models of synergy, the ZIP, the HSS, or low, or the Bliss. All of them score very high. So basically, NCOD 466 and NCPB 916 in cell-based assays, they have a very, very strong synergy. This suggests, if the results can be extended to in vivo models of Mammarenavirus infections, is that by reducing the concentration of each compound, you may be able to alleviate or eliminate entirely possible side effects. And also, you will basically bring the genetic barrier to resistant viruses will be much higher, because for the virus will be very difficult to escape via mutations, simultaneous mutations to both really types of antivirals. And one thing related to antivirals against Lassa or any other hemorrhagic fever Mammarenavirus is that in the case of Lassa, we know that viremia is one of the best predictors of disease outcome. So if the viremia is high, then the outcome is usually poor and is usually fatal. Now, if the viremia is relatively lower, then the individual can recover and control the infection and eliminate the virus. So this is important because what it's suggesting is that in the case of antivirals against Lassa, we don't need an antiviral that can eliminate entirely virus multiplication. It's just to reduce viral load sufficiently to provide the host with a window of opportunity to build up a effective immune response to control the virus and eliminate the virus, which really could be more achievable than trying to have a treatment that entirely eliminate the virus. And finally, let me acknowledge people that they have really contributed to this work. We have collaborated with many different people. All the high throughput screens is done in collaboration with Kristin Johnson at Caliber. All the medicinal chemistry is done by Arnab Chatterjee and Andres Montoya and Chen Wanju as Caliber 2. We have collaborated with Ian Wilson, Andrew Ward, and Stefano Forlì in all the structural aspects that I haven't had the time to discuss. I indicated the collaboration with Matthias Goret at the University of Alberta with the biochemical assays. All the studies with Lassa, with live Lassa and live hunin with highly pathogenic strains that they require BSL-4 have been done in collaboration with Luis Martinez at Texas Biomedical Research Institute in San Antonio. And the studies with hunin were done with Brian Goen at University of Utah. And in our lab, Rufin Show has been involved in all the experiments with animals. Rachel has been involved in all the studies developing MCPP916 as a endonuclease inhibitor of Mammarenaviruses. Hyder Whitwick has been involved in the studies with ENCODE 466, the polymerase inhibitor. And Beatrice Cubitt has been assisting all of them with different experimental procedures. And obviously this was done all under the funding from the National Institute of Allergy and Infectious Diseases, the AVi program.